Hello, and welcome to Chapter 6, all about the 3D structure of proteins. So, up until now, we've been talking a lot about the primary structure, and now we're going to be adding on in this chapter several new levels of protein structure, including secondary, tertiary, and quaternary structure, and then at the end we'll talk a little bit more about how proteins fold. In the second part of this chapter, we'll learn more about structure determination techniques, including circular dichroism, nuclear magnetic resonance, and X-ray crystallography. But first, let's start with the different levels of protein structure. So as we've already learned, the primary structure refers to the amino acid sequence within a polypeptide chain. So it's just the order of amino acid residues that are in that polypeptide chain, and that primary structure is what gives rise to all these other levels of structures that come after it. That's why it's the primary structure. It's going to give the polypeptide a certain propensity to fold a certain way or to behave a certain way. So that brings us to the secondary structure. So a secondary structure for a protein refers to the repeating local structure that's stabilized by the hydrogen bonding between the different parts of the polypeptide backbone. And what's illustrated here on the left side of the slide is one type of secondary structure called a helix that we'll talk a little bit more about in a little bit. The next level of structure is called the tertiary structure. This is a full three-dimensional structure of the polypeptide, and the tertiary structure is stabilized by interactions between the side chains of the polypeptide residue, as opposed to the secondary structure, which is stabilized by hydrogen bonding between the different parts of the backbone. So the example given here for tertiary structure is one subunit of hemoglobin, the beta chain, and that contains a heme molecule in the inside of it in red there. So finally, the last level of structure we have is called the quaternary structure, and this structure is formed by interactions of multiple separate polypeptide chains. And these are also going to be stabilized by the side chain interactions. It's just from multiple separate polypeptide chains that are not fully connected to one another. So only proteins that have, for example, different subunits are going to have quaternary structure. It's also called an oligomeric protein. Oligomer meaning, some kind of mer, like a dimer, a trimer, whatever mer, a tetramer. So oligomer just means a few pieces. All right, so let's move on to discuss a little bit more about each of those types of structures. So we'll start with secondary structure. There are four most common secondary structures that we'll be talking about in this class. There are also some other ones that we won't discuss here. The first one is called an alpha helix, then there's a beta sheet, and then there's a turn and a loop. So let's go into detail about each of these four secondary structures. First, we have the alpha helix. So the alpha helix might resemble a little bit another helix that we should be familiar with right now, which is the double helix of DNA. But it has some key differences. So for the alpha helix of a polypeptide chain, it is formed by one single polypeptide strand. And something specific to the alpha helix is that it's a right-handed helix. So what that means is, if you follow the backbone in the N to C direction, and you use the thumb on your right hand to point up towards the C and towards C, then the fingers of your right hand will curl around in the same direction as the helix. So in this helix that we have shown here, we can see that the N to C direction is going up the helix as oriented with the screen. So this is, for example, 1N, this is C alpha, and this is the carbonyl carbon. And so you can see that the N to C direction is generally up. So if you were to use your right hand, you would turn your fingers in the same direction as this helix. So for every turn on this alpha helix structure, you can fit 3.6 amino acids. So if let's say we wanted to start right here, here is, so it's an alpha carbon, so one amino acid over is over here, and then the next amino acid over is over here, and then the third amino acid over is about here. So a little bit more than 3 amino acids would get you one full turn up this alpha helix. So the next property of alpha helixes is the pitch, and so the pitch refers to the vertical distance between one exact turn. So this is the pitch, and the pitch is always 5.4 angstroms long. Excuse me, 5.4 angstroms. And that very specific length and angle for the turn is because of the way that the structure is stabilized. So part of that is, so one more feature about the alpha helix that's important to know is also that the side chains are pointing out away from the helix. So in this diagram, the side chains are drawn in purple. And so you can see that these are all sort of pointing out of the plane of the helix, sort of, towards the outside as opposed to on the inside. So like when we think about our double helix of DNA, we have our nitrogenous bases on the inside of that helix, holding the two pieces of the helix together with those hydrogen bonds between the bases. In the case of the alpha helix polypeptide chain, you have your side chains actually projecting away towards the outside of the helix. And so that brings the question of what's holding this alpha helix together. So that is going to be held together by the amino acid backbone. So each amino acid in this alpha helix is connected to the fourth amino acid residue away from there. And it's the interaction between the carbonyls and the amino groups of all the amino acid residues. So one example of this is right here. We have our hydrogen bond. And so we should be able to count that these are going to be 4 amino acids away from one another. So if you count the next one from here, this is the carbonyl carbon of the last one. We'll call that the N. This one is the N+1, nitrogen, alpha carbon, carbonyl carbon. Then we have our nitrogen number 2, alpha carbon number 2, carbonyl carbon number 2. In the back here we have our nitrogen number 3, alpha carbon number 3, carbonyl carbon number 3, and finally we have our amino group number 4 connected with our 0th carbonyl carbon down here by this hydrogen bond. So you see this hydrogen bonding interaction that connects each N+4th amino acid to the 0th amino acid before it. And so that's what really helps the alpha helix keep its shape, is that hydrogen bonding in the backbone. And so that structure is a little bit different from a beta sheet. So let's talk about some of the properties of beta sheets. So they're referred to as a beta sheet or a beta pleated sheet, and you can see from this picture on the right, it really does have these sort of pleats in it that come from these very particular angles that are taken by the amino acid backbone. And so if you have just one strand, that's not enough to define a sheet, but once you have 2 strands or more, that's going to define what makes a beta sheet. And those strands are always going to run directly next to one another. They never cross over one another. The length of the strands can be as little as 3 amino acids and up to about 10, so they're generally not super long. And what holds them together is hydrogen bonding between neighboring strands. And so, again, this is a secondary structure, so the hydrogen bonds that hold it together are going to be between the peptide backbone. And so you can see in this picture pretty clearly, if we look at the bottom here, we have the amino hydrogen on one that's hydrogen bonded to the carbonyl oxygen of the sheet. That's its neighbor. And you can see that going all the way up the sheet in between those 2 strands that are running next to one another. The length of one pleat of the sheet, as shown here, is 7 angstroms. And the direction of the side chains actually alternates between faces of the sheet. So you can see on the top here, these side chains are facing the back. Then on the next one down, the side chains are facing the front. And then these ones are facing the back again, and so on and so forth. And so the side chains are pointing, they're sort of pointing out from the sheet, and they're on both sides. So there are actually 2 subtypes of beta sheets. They're called antiparallel and parallel. So the strands of a beta sheet always run next to one another, but there's two sort of options for how that can work. So the first option shown here is an anti-parallel beta sheet, where, as shown in the figure here, the top strand, the N to C, runs towards the left, and on the bottom strand, the N to C direction goes towards the right. And so these are anti-parallel to one another, a little bit like within our DNA double helix, those strands are anti-parallel to one another. But here, this is a beta sheet. It's anti-parallel, doesn't form a helix, just a sheet. So don't want to confuse anyone, but you can think about that similarity and difference with DNA there. So again, these strands are running in the opposite direction, and they have this really nice ideal hydrogen bonding geometry because these hydrogen bonds between the amino hydrogen and the carbonyl oxygen are very linear, and they alternate really nicely between those angles of the sheet. All right, so how do we then connect together the strands of an anti-parallel beta sheet? So we know that beta sheets are usually going to be formed out of one longer polypeptide chain that's going to have to turn back on itself somehow to allow the strands to run next to one another and in an anti-parallel way, where one is going N to C in one direction, the other is going N to C in the other direction. So one fairly simple way to accomplish this change in direction is called a turn. So if you think of your anti-parallel beta sheet, here's one strand. It's going to go around this turn shape, and then that will allow the next strand to be running anti-parallel to the original one. And so you can imagine a whole bunch of these turns can connect together a whole bunch of these little strands, and that can help make up a nice big anti-parallel beta sheet. So the one quality that characterizes a turn is that sharp change in direction that we see here. And so this is going to only take up about 4 amino acids. And the 4 amino acids have to allow for that sharp change in direction. So one way that they keep that really specific, fairly sharp turn is most of them are going to contain a stabilizing hydrogen bond, and that will kind of help pull the pieces of that curved piece more closely together. And in addition to that, there are still going to be some unusual dihedral angles within this turn, meaning the angles between non-consecutive bonds are going to be relatively extreme compared to when bonds are in just a beta sheet or an alpha helix. And so because we have this requirement for such unusual angles, to avoid clashes, we actually see 2 particular amino acids very commonly within a turn that will help allow for that change in direction, but sort of for different reasons. So one common one is proline. So proline, as we should hopefully know from a couple of chapters ago, has a very unusual property in its side chain because it bonds back onto the amino acid backbone, and so that gives proline already a fairly angular shape that makes it relatively rigid. So it's rigid, it's angular. And so if you found a proline inside of a turn, that might help keep the turn in its shape and not be as flexible or as able to rotate away from that turn. Sort of for the opposite reason, we often see glycine in turns. So glycine is very flexible because its side chain is just a hydrogen, right? So glycine has the smallest side chain, and it has a whole lot of flexibility and rotation around its bonds for that reason in comparison to an amino acid like proline. So glycine will allow for that sharp change of direction to happen if you need to, as the protein is holding itself. So let's talk about the other type of beta sheet, which is the parallel beta sheet. So the parallel beta sheet has, again, multiple strands that run adjacent to one another, but here, the parallel one, the strands are all running parallel to one another. So in this example drawn here, they are running from N to C, going from right to left. And so now the strands are going in the same direction, but that means something unfortunate for their hydrogen bonding between the backbones. So because now instead of going anti-parallel, we're going parallel, the hydrogen bonding geometry is not quite ideal anymore. So here we can see that our
our amino hydrogen is hydrogen bonding to the carbonyl oxygen across from it on the next strand, but it's not quite linear. It's close to linear. It's a pretty good hydrogen bond, but it's not quite as ideal as we saw within our anti-parallel beta sheet. So how do we connect together the strands of a parallel beta sheet? So remember, our beta sheet strand now are going in the same direction. So if we have our first one going in to C, we can't simply make a turn like with the antiparallel and go back, because that will make an antiparallel strand that's a neighbor. So the protein has to do something else in order to make a parallel beta strand. And often we'll see that in the form of a loop. So the protein will just loop back on itself to allow the next strand in the beta sheet to be running parallel to its neighbor. And so loops are defined as relatively long and relatively unstructured regions of the polypeptide that you can see in this diagram. They're sort of not forming an alpha helix, they're not forming a beta sheet, they're just kind of hanging out there in space. Their main job is to sort of, at least in this example, is to allow for the beta sheet to form. And so it's going to be relatively flexible to allow for that to happen. And one reason it's relatively flexible is that there's often little or no hydrogen bonding within that unstructured region in the loop. And so there's a lot of freedom of movement in this loop as compared to other more rigid or more highly hydrogen bonded parts of a protein, for example, within an alpha helix or within a beta sheet. And so one of the places you'll see a lot of loops is often on the surface of proteins. Like you can see in this sort of bottom picture here, you have this combination of beta sheets in the core and then some alpha helices around the outside, and all connected by all these loops that are forming on the surface. And so one reason that these loops tend to go onto the surface is because that allows them to interact more strongly with the solvent and hydrogen bond with the solvent. And it could do that in a relatively unstructured or non-permanent, more transient way.
So how do these secondary structure shapes arise? So one way we can think about how these secondary structures form is because they're going to be the most stable, right, for the protein. The protein always wants to be in its more stable, least, lowest energy sort of conformation. And so there are properties inherent to the backbone of an amino acid that helps to lead to these particular secondary structures that we see. So if we look at our amide bond, which is drawn on the bottom of this slide, there's actually a resonance structure. So on the left, we have our sort of typical peptide bond, amide bond that we imagine when we think about amino acids and polypeptide chains. But on the right, you can see that it can form this resonance structure where you have this lone pair of electrons on the nitrogen
that can form a double bond with the carbonyl carbon, and then, of course, this double bond, that electron pair
up to the oxygen. And so essentially you have this transient or this partial double bond character between the C and the N in our amide bond. And so that's going to restrict rotation around this bond, right? You can't have rotation around a double bond. And so because there's always going to be this partial double bond character in that amide bond, we have this restriction of rotation, and we call that the amide plane. And so the amide plane you can think of as consisting of these sort of molecules here, the bonds that are surrounding that restricted rotation amide bond. And so because of this, the bond angles that are in the backbone of a polypeptide are relatively fixed. There are some that are very unfavorable because of this rotation or impossible, and there are some that are more allowable. And so the only conformational flexibility in the polypeptide backbone is only due to the rotation of one amide plane relative to its neighbor. So when you think about a long polypeptide chain, most of the bonds are actually relatively most of the bonds are relatively fixed within a polypeptide chain. But each amide plane that corresponds to sort of each peptide linkage can rotate around the neighboring amide plane. So let's look a little bit more closely at that rotation. So the dihedral angles are what we call what we use to describe the relative positions of those amide planes. And so you can see
in this picture here, the amide plane is represented by this sort of greenish rectangle, and that at the center of that you have that peptide linkage between the nitrogen and the carbonyl carbon that has that partial double bond character. And so all of the bonds surrounding that partial double bond are going to be fixed because of that not allowed rotation around that central bond. And so that allows for the definition of two dihedral angles between the amide planes. So let's go a little bit more into that. So the dihedral angles refer to the angles between non-sequential bonds. So what does that mean? The first one we'll talk about is called phi, which is drawn by this Greek letter here. And so phi refers to rotation around that N2 alpha carbon bond before the R group. So between the first N and the first alpha carbon. And so that's shown in red on this diagram. And so when you're looking for rotation around that bond, the phi angle, you're really looking for the angle between the two neighboring bonds, between the sort of before it, the previous carbonyl carbon connected to that nitrogen that's part of that phi bond, and then the bond sort of after it, the alpha carbon to the next carbonyl carbon. And so if you think about the angle between what's drawn here in yellow and blue, you see about 180 degrees, right? They're pointing sort of exactly away from one another in this particular picture. So you could say the phi angle is about 180 degrees here. The other type of dihedral angle is the psi angle. So that's drawn by this Greek letter. And so the psi refers to the rotation around the alpha carbon and carbonyl carbon bond, so the bond right after the R group. And just so you know what I mean, the R group is shown right here. So that alpha carbon to carbonyl carbon bond is shown in blue, and so you're looking for the bonds right before and right after it to define psi. So the bond right before it is that N2 alpha carbon bond, and the one right after it is the carbonyl carbon to the next amine bond. And so here for the psi angle, you're looking for the angle between that bond circled in red and the bond encircled in yellow. And again, here we see they're about 180 degrees apart. They're sort of facing directly away from one another. And so something to think about as we've sort of been leading up to, not all dihedral angles are possible for a few reasons. So one reason is essentially because of things like steric clashes or just repulsion. So on the bottom of this slide here, we can see the impossible dihedral angle because of the backbone itself. So you can see the lone pair from the amine nitrogen and the lone pair from the carbonyl oxygen are just getting in each other's way. They're repelling each other, and so the backbone of the polypeptide just can't form that angle. It's not possible. Another reason, as shown more on the top here, is when you have large and bulky side chains. So side chains can physically get in the way of some types of bond rotation, and so that will make that particular dihedral angles impossible sometimes, depending on which side chains are near each other and how those side chains themselves might like to interact with one another. So getting back to those allowed dihedral angles, we can draw what's called a Ramachandran plot, which we see here on the right, and that shows which dihedral angles are allowed, and it also tells you what secondary structures correspond to that set of dihedral angles. So in blue on this plot are the most favorable dihedral angles, and we can see some familiar secondary structures within those blue areas, right? So you have, and then we can also have in green are also allowed dihedral angles that are a little less favorable than the blue ones. And so pretty much all of the secondary structures are found within these blue and green islands. So circled in red here is the anti-parallel beta sheet. And you can see here that the angles in an anti-parallel beta sheet are just about at negative 180, which is the same as 180, and positive 180. And so that's sort of allowing for the groups to be pointing sort of the furthest apart from each other, and that more sort of ideal interaction between the amide planes. Just underneath that, we have in yellow the parallel beta sheet. So those angles are a little bit less than planar, right? So if it's all 180 degrees, we're all planar. So an anti-parallel beta sheet gets us the closest to planar. The parallel beta sheet is a little bit less planar because the angles are not quite as close to 180 there. And then in purple, so on the other quadrant of our plot, we have the right-handed alpha helix, which is still a very favorable structure, but now we're seeing we're not planar. And as we know, alpha helices are not planar in shape, so it makes sense that they're not near one of those corners. The other structures drawn here that we're not going to be discussing in this class, there's the alpha L, that's the left-handed alpha helix, and then C is for coil-coil, but we won't be stressing those in this class. So that covers what we wanted to know about secondary structure. So now let's talk a little bit more about tertiary structure. So tertiary structure refers to the entire 3D structure of a polypeptide chain. It includes the composition and arrangement of secondary structure elements. And so here in this picture on the right, we have a bunch of alpha helices over here that are surrounding a prosthetic group, maybe a heme. We see a couple of beta sheets over here that are sort of interacting with one another and connected by, looks like some loops or turns. And then on the bottom we have a mixture. We have some beta sheet character and some alpha helix character. And so the composition and arrangement of all these secondary structure elements is what gives us our tertiary structure. And so tertiary structure, unlike the secondary structure, is stabilized by side chain interactions. So it's the different side chains. So for example, if you have, two different alpha helices, remember the side chains are pointing to the outside. So if you have two different alpha helices close together in space, now you can have interactions between the side chains of alpha helices. So for example, this yellow one and this green one, all the way on the right here, something can hold those together, right? You can have salt bridges, you could have hydrophobic interactions, you could have hydrogen bonds. There's a number of, based on which R groups or which side chains are present in the primary structure, that will tell you which types of interactions are going to help stabilize all those different, holding the different secondary structures together into a tertiary structure. And so those interactions, like I mentioned, include hydrophobic interactions, hydrogen bonding, salt bridges, disulfide bonds. Depending on what R group you have, you can have any of those happening within a tertiary structure. And so this is, of course, highly complex and varied, depending, of course, on the primary structure of the protein, which itself can be extremely variable. And then also depending on which secondary structure is formed, and then how all those secondary structures are interacting with one another, we're just adding layers of complexity upon all the complexity that was already there. And so the tertiary structure can also include non-protein structures. For example, in this picture, we have what looks like a heme group that might be inside of a protein and helping the protein either have its function or keep its shape. And so so non-protein structures can be included as part of the tertiary structure. Another component of tertiary structure are motifs. So motifs are commonly observed patterns of secondary structure elements. And let's go into a few more examples of those. So one common motif is called the beta hairpin, and this is just an anti-parallel beta sheet that's connected by a bunch of small loops. And so we'll call that a beta hairpin. It's a number of beta sheets and beta loops, or a beta sheet with a bunch of loops. When you have enough beta hairpins, if you keep adding them onto one another, you can get what's called a beta barrel. So essentially the sheet goes all the way around and kind of loops back on itself, and it actually makes a sort of cylinder or a pore shape that can help, and sometimes items can pass through it, which is shown in this example here. We have this small molecule that's passing through this beta barrel, or somehow interacting with the middle of it. Another common motif we see is the beta-alpha-beta motif, and so this is when you have a parallel beta sheet, and we talked about how parallel beta sheets can be connected by loops. Here we actually see it connected by an alpha helix. So you have one beta strand, and then in order to turn it back around for the next strand to be parallel, there's an alpha helix in between, going back to the beginning. And so when you string enough of these together, you can get what we call an alpha-beta barrel. And so you can, which ends up sort of looking like a beta barrel on the inside, which you can sort of see here in the structure, surrounded by alpha helices on the outside. So all of these different motifs are commonly observed, and they can help make up the total tertiary structure of a whole protein. Something else that's part of tertiary structure is called a domain. So domains are globular regions of a protein. The one in this example on the right, we have a protein that has two domains. There's this blue N-terminal domain that seems to have one function and binds with one molecule, and this C-terminal domain that makes a different shape and might carry out a different function. So domains are usually found in proteins that have greater than 200 residues. And the domains tend to fold somewhat independently of one another, even when they're on the same polypeptide chain that's part of the same, tertiary structure. There could still be individual domains that might fold separately and carry out different functions. So the example that we have here on the right is a protein called glyceraldehyde 3-phosphate dehydrogenase. And like I hinted at, the N-terminal domain binds NAD+, that's in blue. The C-terminal domain, that's in orange, binds GAP. And so there's two different domains. They're part of the same protein. They're in the same long polypeptide chain. But as part of the tertiary structure, there are two different domains that each carry out different functions. And a lot of times during evolution, gene duplication can occur at this domain level. And so we've discussed how, for example, paralogous proteins, you might have gene duplication that results in two different proteins with a common ancestor that might carry out different functions. This can also happen with domain. You can have duplication of a single domain from one protein that can maybe add itself onto a different protein. And so you can sort of swap around these functions or sort of trade different functions among different proteins within an organism, which is kind of cool. The last level of structure we're going to talk about here is the quaternary structure. So the quaternary structure is the interaction between polypeptide chains or subunits of a protein. So up to tertiary structure, we're talking about one long polypeptide chain. Quaternary structure is when we have multiple different polypeptide chains, which we often call subunits of the single protein. So there can be a number of different subunits within a quaternary structure. There can be just a monomer, that's one polypeptide chain, dimer, trimer, tetramer. In our example here on the right, we have hemoglobin. Hemoglobin is a tetramer, as you can see with those four different color subunits, the dark blue, the cyan, the light green, and the yellow. So we call that a tetramer. And whether or not the subunits are the same to one another helps give us a prefix for this. So we can refer to the quaternary structure as homo if all of the subunits are the same as one another, or hetero if they're different from one another. So in the case of hemoglobin, it's hard to tell from this picture, but there are actually two copies of two different subunits within each hemoglobin quaternary structure. So the tetramer hemoglobin has two of one subunit, two of a different subunit. So because not all the subunits are the same, we would call hemoglobin a heterotetramer. And so the overall structure can contribute to the quaternary structure, including the arrangement of the subunits, the types of interactions between the subunits. So again, we're talking about side chain interactions here. So hydrophobic interactions, hydrogen bonding, dipole interactions, maybe even disulfide bonds. And non-protein groups can count here too. So here in hemoglobin, the little red part that you can see within each of the subunits is actually a heme group. And so non-protein groups like heme can count within the quaternary structure of a multiple subunit protein like hemoglobin. So overall, if you were going to talk about hemoglobin, as a quaternary structure, we call it a heterotetramer with 4 heme groups. So that brings us to a little bit more about protein folding. So here you see this sort of funnel-shaped diagram that depicts the energy and the entropy as a protein is folding. So if you think about sort of the beginning here on the upper left, this is when the protein is completely unfolded. Let's say it's a brand new protein, it just got translated, it hasn't found a shape yet, or for whatever reason it got unfolded. So here it has fairly high energy, and it's fairly disordered, so it has also high entropy. And so the protein naturally is going to try to find a lower energy shape that also causes it to be more orderly and therefore also decrease entropy. So it'll naturally start to form these secondary structures that we just discussed, like beta sheets and alpha helices. And as it does this, the energy level will start to go down as it finds these more stable shapes within the larger protein. And then these shapes will keep folding on each other and sort of building up the larger and larger overall shape of the protein until finally you get to the native structure, which is at the bottom of this funnel here. So the native structure is the most orderly, and it's also the lowest energy, most stable shape for this protein. And it's sort of this very narrow place that's sort of hard to get out of once you're into, because it's so stable and so orderly. But issues can happen as a protein is folding. You can see here that this is not sort of a smooth line going down, right? You have these valleys next to these slightly higher energy need to get out of them. So these are sort of what we call local minima for the energy, where if a protein happens to sort of, let's say a protein is folding, right? It's making its way sort of down this funnel. It gets past, it goes to here, it goes to here, but then maybe somehow it ends up making a set of folds that get it to here, which is relatively stable. It's a local minimum for energy. And so proteins can actually sometimes get stuck in these sort of partially folded, semi-stable states. And sometimes they'll need the help of a chaperone protein or other kind of help to actually get it over this bump and bound to its fully native conformation. So that brings us to, how does the protein find the sort of stability as it's folding and finding its shape? So one way it does this is, as we talked about for tertiary and quaternary structures, the side chains are really important, because the side chains interacting with one another help to both help the protein keep its shape, as well as help the protein interact with its environment, the solvent that it's in. And so we have this hydropathy scale, where the most hydrophobic side chains have a higher degree of hydropathy, and the more charged amino acids have the lowest degree of hydropathy. And so you'll see for the more hydrophobic residues, they are much more likely to be buried in the core of a protein, or possibly within a bilayer membrane where they can sort of hide from solvent. Because these hydrophobic side chains are not going to want to interact with aqueous solvent. And so it's more stable and more lower energy for them to hide from the solvent. Compared to these more hydrophilic residues, these are more likely going to be on the surface of a globular protein, or in some way exposed to the surrounding solvent, because they are going to want to interact more with an aqueous solvent, and overall, that will give the lowest energy, most stable shape for a protein. So something to keep in mind with protein folding is that it is reversible, and this is a technique that sometimes we'll use in the lab. And so when you have a native protein, as shown in this diagram, that has this sort of correct conformation, it's active, it can perform a function, it will have a particular shape made of a secondary and tertiary structure, as well, that's holding, that's mostly, the shape is mostly held together by non-covalent interactions, right, like hydrogen bonds and hydrophobic interactions. And so one chemical you can use to help sort of just unfold a protein is called urea. So urea helps to unfold a protein structure, and it does this by disrupting non-covalent interactions. The other thing you'll need to do to unfold many proteins is reduce disulfide bonds. So disulfide bonds, as we know, are covalent bonds. Urea is insufficient to disrupt disulfide bonds, and so you'll need a reducing agent like beta-mercaptoethanol, or BME. And we learned a couple of chapters ago how BME can be used to reduce disulfide bonds. And so once you've added urea and BME, you've fully denatured or unfolded this protein. And so if you want to refold that protein, then you can just remove the urea and the BME. And if you do that, you'll follow along this side of the path, you'll return to this native or active conformation for this protein. If you remove the urea and the BME at the same time, it will want to find its minimum energy and most orderly state, and it will refold. But something interesting can happen actually if you only do that, if you take away the urea and BME one at a time. So if you only, and now we're going in this direction, so if you only remove the BME from your denatured protein, the protein is able to start forming disulfide bonds, right? Because it's only in the presence of urea. And so, but those disulfide bonds might not necessarily be in between the correct cysteine residue. And so you can get what's called a scrambled protein, where the disulfide bonds have formed the wrong way, which leads the protein to be unable to get back to its correct shape, even if you do remove the urea. So you have a problem, right? So how do you fix this problem where you have a protein that wants to fold, but it can't because these disulfide bonds form that are forcing the protein into this incorrect shape? What you can do there is add just a small amount of beta-mercaptoethanol, and you add just enough to break some of these disulfide bonds, and then because now there's no more urea and the protein wants to make its native shape, the disulfide bonds will reform correctly, and they do this oxidation process. I forgot to mention this, but to reform the disulfide bonds, they have to be oxidized, right? And the oxygen that's present just within the air or within, dissolved in a buffer is sufficient. So one example of protein folding, or I should mention that protein folding is also a trial and error process, right? So as we sort of looked at with our picture here, the protein has a lot of options along the way. It's sort of trying to fold, maybe it finds a local minimum, maybe it tries another shape, comes out of it, finds an even lower minimum. And so you have this, it's not necessarily a linear journey, the protein is trying out a number of different shapes until it finds its best native structure. And so there's a little video of what that looks like when we're talking about refolding of GFP. So let's take a look at this video. And so you can see that the protein is trying out all these different shapes, right? All the dihedral angles are changing against one another, and the shapes that are most stable, the most highly stabilized by hydrogen bonds between the backbone, in the case of a secondary structure like this beta barrel that you see here, and that alpha helix that you can see forming, and then compared that to, so like you can see the formation of these more stable secondary structures between the hydrogen bonding of the backbone, as well as those sort of loop regions in between the secondary structures that are maybe more disorderly, that might interact more with solvent. So that brings us to sort of a more clinical connection. So we just saw a video of GFP folding correctly, but there are a number of diseases that we are aware of that are caused purely by protein misfolding. So one example we have of that is prion proteins. So prion proteins are naturally found in your brains and your neurons. And so they have some sort of normal function when they're folded correctly, but we're actually not sure what the prion protein's native function is to do, but we all have them in our brains. And they normally have, on what we see on the left, the shape where you have an alpha helix, primarily an alpha helix shape. But there's a switch that can happen where a misfolding happens that you can see on the right. And as the prion protein misfolds, it starts to form this beta sheet. And the beta sheet is a highly stable structure, right? We talked about that. And so the more of these beta sheets you get, the more, and once you have one sort of misfolded prion protein, it's almost infectious. It can interact with another prion protein. Maybe that original prion protein was normally folded. If a misfolded prion protein interacts with a correctly folded prion protein, it will cause it to become misfolded. And so in that sense, the prions are almost infectious because they'll cause the other normal prion proteins to become misfolded. And so ultimately, if you have enough buildup of those, you can get damage to your neurons, and you'll get almost holes in your brain tissue, which is why one of the diseases it's called is called bovine spongiform encephalopathy, or BSE, mad cow disease. And when humans get it from eating, for example, infected meat, it's called Creutzfeldt-Jakob disease. And obviously, it's an awful neurodegenerative disease. And so another neurodegenerative disease that we can see that's also due to protein misfolding is Parkinson's disease. So the alpha-synuclein protein is also normally found in your brain. It's normally an alpha helix shape, and it's normally found on a membrane. But if it comes off the membrane for whatever reason, when it's in solution, it's more unstructured, as you can see in that next picture. And so when you have enough of these unstructured alpha-synuclein proteins, you end up forming this sort of unknown oligomer, and that oligomer can arrange itself into these beta sheets, which ultimately are one of the components of amyloid fibers. And so it's thought that these oligomers, these number, dimer, trimer, tetramer, some small number of polypeptides that it forms the beta sheet, is toxic, especially to your dopaminergic neurons and the signaling through that pathway. And so that is one of the causes for Parkinson's disease. So now that we've gone through all these different levels of protein structure and some clinical connections, please go ahead and answer the practice questions.